When tissues need to eliminate damaged or dangerous cells, they typically do so through one of two distinct physical pathways. On the left is classical apoptosis, characterized by a neat, controlled shrinking and membrane blubbing. On the right is lytic cell death, where a cell violently swells until its outer membrane ruptures, spilling its internal contents into the surrounding tissue. For decades, cancer biology operated under a single dominant theory. Tumors survive primarily by evading apoptosis. This led to a massive push to develop drugs that restore the apoptotic pathway. In liquid tumors like leukemia, targeted molecules like BH3 mimetics successfully strip away anti-apoptotic defenses, forcing the cancer cells to finally shrink and die. But in solid tumors, restoring apoptosis routinely fails. Malignant tissues quickly evolve resistance to the blunt force cytotoxicity of standard chemotherapy, creating populations of drug-tolerant persister cells that simply shrug off the damage. The field is now shifting its focus from the evasion of apoptosis to the evasion of cell death at large. Researchers have mapped entirely new, non-apoptotic modalities of regulated cellular demise, specifically necroptosis, pyroptosis, and ferroptosis. Achieving complete tumor clearance requires an approach that steps past broad toxicity. To defeat treatment-resistant cancers, we have to weaponize the exact physical mechanics of how a malignant cell breaks apart. Destroying a cancer cell is only half the battle. The long-term efficacy of any cancer therapy relies entirely on the immunological consequence of its death. How it dies dictates how the immune system responds. We can measure this response using an assessment model known as the Immunogenic Cell Death, or ICD, Triforce. This matrix tracks three pillars: antigenicity, which is the presentation of tumor antigens; adjuvanicity, the release of damage-associated molecular patterns, or DAMPs, that act as alarm signals; and inflammation, driven by specific cytokine and chemokine profiles. Depending on how these three variables align, the tumor microenvironment generates a specific output. This ranges from a completely tolerogenic or immune suppressive state on the left to a highly immunogenic T-cell activating state on the right. By mapping the molecular pathways of distinct cell deaths against this matrix, researchers can precisely calibrate the tumor microenvironment, engineering the tumor to act as an endogenous vaccine against itself. The baseline for this comparison is apoptosis. The intrinsic apoptotic pathway initiates when internal stress forces the proteins Bax and Bach to permeabilize the mitochondrial outer membrane. This structural failure unleashes caspase-3 and caspase-7. These executioner enzymes systematically dismantle the cell from the inside out, neatly packaging the cellular debris into sealed vesicles for macrophages to silently clear away in a process called efferocytosis. Returning to the Triforce matrix, apoptosis plots neatly on the far left. Because the cell preserves its antigens, antigenicity is high. But because the plasma membrane remains perfectly intact, no alarm signals or inflammatory cytokines escape. The adjuvanticity and inflammation sliders remain firmly at zero. Apoptosis goes a step further and actively suppresses inflammation. As caspase-3 and 7 tear through the cell, they physically cleave and disable the CJAS sting pathway, the exact molecular signaling line that would normally trigger an immune-stimulating interferon response. Inducing apoptosis in a solid tumor generates a silent, tolerogenic void. Without danger signals, local dendritic cells are skewed toward activating regulatory T cells, which suppress the immune system and protect the surviving cancer cells. Necroptosis offers a stark mechanical contrast. This is a programmed lytic demise that ends in plasma membrane rupture, driven by tension-sensing proteins like Siglec-12 that physically tear the outer membrane apart. Necroptosis is tightly regulated by a molecular brake called caspase-8. The pathway only engages when caspase-8 is functionally inhibited by a drug or mutated by the tumor itself. Once that brake is removed, an execution cascade begins. The kinase RIPK1 autophosphorylates, recruiting RIPK3. Together, they form a complex that targets and phosphorylates the terminal effector protein, MLKL. Activated MLKL physically changes shape, allowing it to lock together into oligomers. These protein structures traffic directly to the plasma membrane, driving themselves into the lipid bilayer to form massive ion-conducting channels. The osmotic balance collapses, fluid rushes in, and the cell violently bursts. Plotting necroptosis on the ICD triforce shifts the output entirely. As the membrane explodes, massive danger-associated molecular patterns and cytokines flood the microenvironment. The adjuvanticity and inflammation sliders max out, driving the local environment deep into the immunogenic zone. This eruption provides the precise chemical adjuvanticity needed to prime cytotoxic CD8+ T cells, transforming immune-evasive cold solid tumors into highly targetable hot tumors. Pyroptosis operates with similar violence. Originally evolved as an innate defense against pathogens, it is driven by internal sensors called inflammasomes and activated by specific enzymes like caspase-1 or granzymes delivered directly by attacking T cells. The terminal executioners of pyroptosis are the gasdermin protein family. When a caspase cleaves a gasdermin molecule, it cuts away an inhibitory domain, liberating the protein's N-terminal. These liberated terminals insert into the membrane to form massive, ring-like pores. Through these massive pores, the dying cell vents a highly specific inflammatory payload, deploying potent amplification cytokines known as IL-1 and IL-18 directly into the surrounding tissue. Looking at the triforce matrix, pyroptosis sits cleanly alongside necroptosis in the immunogenic zone, characterized by a massive spike in IL-1 and IL-18 data nodes. It acts as an ultimate innate immune alarm. Because this alarm aggressively recruits cytotoxic lymphocytes, solid tumors frequently survive by epigenetically silencing their own gasdermin genes. This active evasion is driving an intense pharmaceutical push to discover drugs capable of reactivating gasdermin expression inside malignant cells. Ferroptosis sits completely apart from the other pathways. It lacks a canonical receptor or a dedicated protein pore. Instead, it occurs when a cell trespasses a lethal threshold of iron-dependent lipid peroxidation. A cascading wave of oxidation rapidly destroys the cell membrane. Rapidly dividing cancer cells require immense metabolic flexibility, which generates heavy oxidative stress. To survive, they rely entirely on chemical surveillance safeguards, specifically the system XC to GPX4 axis and the SPF1 axis, to constantly neutralize lipid peroxides before they accumulate. When these safeguards are pharmacologically blocked, the tumor undergoes ferroptosis. But rather than releasing immune stimulants, ferroptotic cells leak oxidized lipids, aldehydes like 4-HNE, and immunosuppressive molecules like PGE2. On the triforce matrix, ferroptosis reveals a strict duality. It fluctuates wildly between tolerogenic and inflammatory, depending on the exact stage of the tumor. The release of PGE2 actively suppresses the recruitment of dendritic cells, essentially blinding the immune system to the dying tumor. Ferroptosis represents a profound vulnerability for metastasizing cancers, which require highly oxidizable lipid membranes to move through the body. But implementing ferroptosis-inducing drugs requires exact spatial and temporal precision. Otherwise, the therapy risks sabotaging the patient's own adaptive immune response. These modalities do not exist in isolation. They form a highly interconnected, competitive network. Blocking a signaling node in one pathway forces the biochemical flux into another. Cancer cells manipulate this interactome to create dead ends. A tumor might hypermethylate the RIPK3 promoter to disable necroptosis, or mutate caspase-8 to shut down targeted apoptosis. To bypass this, researchers are designing sequential therapeutics to hack the network. First, they apply an interferon-inducing therapy, like a STING agonist or targeted radiotherapy. This forces the tumor to upregulate and rebuild its missing necroptotic machinery. With the machinery rebuilt, the patient is given a SMAC mimetic combined with a caspase inhibitor. By intentionally disabling the apoptotic fallback, the entire cellular signaling flux is forced into necroptosis, triggering a violent, highly immunogenic collapse of the tumor architecture. The future of precision oncology relies on moving past broad-spectrum DNA damage. We must focus explicitly on the molecular architecture of how a cell breaks down. By mastering the crosstalk of the cell-death interactome, medicine can force a tumor to provide both the antigen and the chemical alarm. The cancer itself is engineered to instruct the immune system, turning the disease into its own cure.